Yeah.
So again, welcome everyone to our bioelectrodynamics webinar. We have it recorded now. It's being recorded. And I would like to welcome today our speaker, Rui Wang from Foshan University. It's my pleasure to have you here. And before we start, I would like to give you just a few housekeeping comments. So uh, in our settings in the Zoom, we have that attendees are muted and have their screens off. However, you can use the chat or Q&A session here to ask the questions, to type your questions, and then after our speaker is finished with his talk, we'll take the questions, and also I can allow you to get unmuted and you can pop up, show your video and ask the questions yourself. So that'll be after the talk. So talk will take roughly 45 minutes. And then we have the Q&A session. I would also like to thank to our supporters. Our webinar is technically sponsored by International Union of Radio Science, URSI, and also by Engineering in Medicine and Biology chapter of the Czechoslovakia section of the IEEE. Now I would like to briefly introduce our speaker. So Dr. Wang is an exceptionally young researcher from Foshan University, and he graduated from South China University of Technology, and he's been publishing very interesting papers in a variety of top food technology and chemistry journals. And he's interested in the both mechanisms of the pulse electric field effects on proteins, particularly plant proteins, for their modification and for application in for example, degradable packaging or quality improvement of fruits and vegetables. So with this, I would like to give word to our speaker. Please go ahead. If you can make it full screen, we can start your presentation. Please go ahead.
Okay.
Yes, perfect.
Uh, it can be.
Yeah, we can see your screen.
It,
Everything is fine. Everything is set. You can go.
also, so everybody is come here. Is come.
So, yeah. So feel free to start anytime if you are ready.
Uh, okay, Michael. Professor Chen told me that he also want to join today's
Oh, okay.
sharing,
Very
but
good.
he's in the subway. Maybe not easy to
No
in here.
problem. No
So
problem.
he want me to tell you that he want to join, but maybe this time cannot. But for last
No
time he
problem.
want sharing. Yeah,
It's very much appreciated that the
yeah,
professor
yeah.
also mentioned that.
President
Okay, I hope we
just
will have
call
him.
me by the phone. Yeah, tell me that.
Very good. I hope we will have him as a speaker in the next
Yeah, yeah,
occasion, the beginning of next year. But please now
yeah,
go ahead, Ivar talk. You can just make a full screen and start.
yeah.
So is everything okay?
So, so I can beginning of
Yes,
today's.
yes, please start.
Please.
We
Okay.
are waiting for you.
I think maybe not so so everyone is coming the Zoom.
Yes, we can see the
Yes.
screen. Just please make it full screen and you can
Okay,
start.
okay. So the screen can we see?
Yes,
Yeah.
yes, you can.
Okay, okay, okay. Okay. Hello everyone. And today, my name is Wang Rui. You can call me Wang. And I'm from Foshan University. And it's my pleasure to be here to introduce my research. And and thanks for Dr. Michael giving me the opportunity to be here. So the research is focused on the PEF technology in plant protein modification. But actually, for my research title is about the effect of PEF combined with pH shifting treatment on the aggregation structure and functional properties of soybean protein isolate. So I want to focus my research, and maybe from my research, I want to you know that soybean protein isolate maybe is one of the important plant protein, and so I want to share this title. Now I will short in soil protein isolate as SPI, and plus the electrical field to be PEF. It's okay. 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 I'll show in my uh, showing my content. So I have four chapters. Chapter one is about the background, and chapter two is what I have do and the results. And chapter three is the conclusion of my result, and chapter four is the perspectives of what we will maybe have the cooperation or what we will do in the future. Now, let me for 1 minute to introduce myself. As you know, my name is Wang Rui, and I'm from working now. I'm working in Foshan University, and my research areas. One is the plant protein modification by PEF, and for the packaging and non-soluble processing of food. And I'm graduated from South China University of Technology in China, and I have been published for 13 science 13 SI paper with a total of 1,020 and for CITES and index in of 21. This is my just a very small um, very small introduction. So I will begin today's research talking. First, I will share the background of what I have do, as well as that plant soil pot sources. The potting has the chance to replace the animal soil seed pottings because the plant soil pottings have some advantage like renewable and eco-friendly, and also the price is very low than it can be more cheaper than the animal pottings. But for animal pottings, the big problem is that it cannot be, it's non-renewable, and maybe during the animals maybe have the greenhouse gas, maybe carbon dioxide oxide, and one problem is the religions. So maybe I cannot um, focus on the religions to talk here. So now we want to choose the plant soil seed pottings to replace animal soil pottings from this advantage. But and so I want to talk about in food, what kinds of plant proteins can be see. You can see these pictures. It can be classified as this three major sources, and in food industries, the common used in food industries is soybean protein, maybe SPI, and the peanut protein in food chemistry, in food industries, and the rice protein in food industries. In food industries, the protein can be used as the food additives. You know that can be used to be the foam foam engines because the protein have a very good plant protein have a good foam properties, and another and can be used as the food additives in the food industry. And another thing is that plant protein have is a nutrition have many nutrition the view that can be used in the food industry, but for the plant protein has some disadvantage. Then maybe I will focus on three advantage. So first is the problem is the energy, because peanuts and soybean protein maybe cause the enrichments to our bodies. And this is the first problem. And the second problem is that the low absorptions, because here I want to share very interesting stories, because I have a student who like exercise. He exercise every day, so he do experience by himself. He eat two kinds of protein powders. One is from the milk, WPI, and one is from the soybean, means SPI. So he eat these two kinds of protein powders for 2 months. Everyone for 1 month. He found that he eats the milk protein, maybe the milk protein powders. He feel the energy, and his muscles groups very quickly. But he eats the plant protein. He feel tired during the exercise, and the muscle cannot grow very quickly than the milk protein milk protein powders. So this is because of the low absorptions of the plant protein. And then the third problem is that in maybe focus on our food industry, means that when we eat some plant protein foods, we maybe feel rigid textures and poor flavors. Because that for the rigid textures is because that plant protein is difficult. Many plant protein is difficult to be to be in the water, because the solubilities of plant protein is not very well than the milk protein. This is one problem. And the second is the poor flavors, because SPI, soybean protein, maybe have a bitter flavor. So this in the food industries, we need to solve these three issues. And I focus on is about the road textures, about plant protein not have a good solubilities in the water, maybe cause the road textures. So first, we talked about the road textures of plant protein is associated with their low solubility. Why plant protein have a low solubility? Because of their stable rigid structure. As we can see that because today I will sharing about soybean protein SPI. For SPI, you can see they have two main components. The SPI is a mixture proteins, not just one protein. And the two main components is 7S and 11S. And we can say that this protein structure is very rigid and stable, and show a very low flexibility. So the water molecules is difficult to contend with to interactions with these proteins. So this protein because of their structures is very rigid, so it cannot be easy to in the water, and maybe show aggregation behaviors in the water. So I'll talk about the SPI aggregation. Is that we get the SPI in food industries way from the soybean. Yeah, soybean is rich in SPI, but actually we want to be honest, we want for the soybean, we want what do we want to extract is the soybean oil, yeah. But for SPI, is byproduced, and byproduced then maybe contain 15% SPI. So in the food industries we will extract of the SPI by the byproducts and during the soybean oil and to give. I hope you can understand what I'm talking. Yeah. Then how to get the SPI? To get the SPI soybean protein isolate, we need to extract the protein by the by this by a very easy, and I think it's a very easy methods is alcohol solutions and acid insulation. You know in the acid insulations we can get the protein, and the protein actually is actually in the lab. In the lab we just use the get the protein is in the water, but we need to be a power for less maybe analyzed or for the less in for the industries application. So you need to freeze drying. So maybe have two methods to drying. The first one in the lab is the freezing drying. You know that just in a very low temperatures to get the power of the SPI, and this methods have advantage in that can not break the SPI structures, the orange structures of SPI, and the water solutions maybe have higher solubilities, maybe about 13 to 18%. But for the spray dining, in the industries, we need to kill the enzyme. We need to kill the enzyme because and we need to sterilize of the and there's two reasons. So we must use the spray dining, but spray dining, very high temperatures, maybe 113 and 13 and these higher temperatures. So maybe this time if we use the in food industries, and this got the powers maybe have not get a very good solubilities because the denaturation and aggregation of the protein may be just 20 to 13%. So SPI aggregation structures, what they will maybe very affected by the temperature or by the pictures or by the oil concentrations. And for 7S and 11S, they will combine with, or interactions to the hydrogen bonding and hydrophilic interactions to buildings, and finally to form the dense aggregation structures. And at this time, these structures, proteins, maybe show very low solubility and very poor functionalities and limited application in the food industries. This is the main problems about soybean protein isolate during the food processing. So, the aggregation of the protein of the plant protein of SPI, how to modify it. Here we will talk about one of very interesting methods is about the flexible methods. The flexible method processing is how to do, because for the region errors is difficult to break, but actually the protein out of protein, you can see that they have the flexible arrows. So if we use some methods to modify the flexible arrows and make a protein to folding, and maybe you can to be from the regional structure to be flexible structures, maybe proteins, maybe the aggregations behaviors can be changed and maybe have a very maybe in the functional enhancement will have a very high level, maybe that the functional may be improved. So this is our PEF can do, and another technology can do just is flexible processing to the SPI or the plant potting. So the modification methods in the plant potting may be a lot plant potting for all the potting, maybe milk protein of animal proteins can be classified as three methods: the physical and the chemical and the enzymatic modification. For the physical, the traditional method is a heat, just a heat or serum or heat for 30 minutes or maybe a higher temperatures. But these methods have some disadvantage. Long time, maybe 30 minutes or 1 hours, and high energy and maybe sometimes maybe have very limited modification effects. But actually, for ultrasonic or ultra high pressures, maybe we can show it in US with UHP and PEF. Maybe this method is the have advantage of short time and high and maybe more green and more eco friendly. But for this is why we chose by PEF. And for chemical modifications, the problem that the chemical regions in the fruits may be affecting the safety of food, and for the enemy and a high cost because many enzyme maybe have a very a very high price and then cannot in the food industries. Cannot be used in food industries. Actually, if we need to very to have a very high or very ranged application, just use the enzyme is cannot get. But for physical modification, for PEF is okay. So how about PEF? As we and although everyone know PEF, but PEF in food industries can do what how many things they can do. First, they can enhance the nutritions. The plant soils. Maybe have some very bioactivities active compounds will be restrictions from the BioPEF. And second is about the enhanced dining. I forget in which countries they called other companies have the machines to drying for the strawberry and to make the strawberry have a very good shape. And another thing is in food industry, for the milk sterilizations, and this is a very big apply. I think it's very high, very range application in the milk and another bill like wine to sterilizations, and the last one I think maybe the modification. For the starch, for the potting, for maybe for the manicures, it can be used the PEF to modify it. So how about the PEF for potting? And this pictures is also from some reviews, but I'm sorry that I not list the reviews here. But another PPT will share the reviews here. Okay, you can see that when we use the out of the electric fields applied to the outside of the potting with the dipole moments angles or polarization polarization reactions. The polarization reactions is many reviews talk about. And the porting will have change, that maybe open the maybe open the structures to be unfold and to be a random coin, in their secondary structure. And then change and finally change the porting structure. Just use the forces of electric field. So this is a background of our research. So I will sharing my research results from in here. So how to preparation of our SPI. You know that SPI was so for time I will dissolved in the water to a final concentration of 10 and processing in a continued PEF processing equipment. And during the equipment, we maybe for the PEF we can face the plus ways of pyrolysis and the frequencies, and maybe and we focus on the processing time. Actually, for focus on maybe we in a processing time, but we will varying the PEF field strength. And finally, the SPI solutions as and will be collected and for the further analyzed by some physical structure analyzed, and the solubilities and the molecular weight and the structures and the solubilities. And this is our groups, the machine. You can see that we just use the continuous processing system, just use these machines to pump the water from the to the system, PEF system, to treat it by PEF. So I will show you the results. Firstly, we can see from this picture that we can see that during after the PEF treatment, we can see that in the 10 in the tens, we can see that the solubility have a very high price of 77%. Then the orange maybe have 17. So this maybe shows that at the medium field strength, the solubility can be increased, and we use the turbidity and particle size descriptions to analyze the aggregation behaviors. We found that with in the medium field strength, the aggregation behaviors of the orange and protein was improved. But with the increase of the PEF strength, we can see that at 13, you can see that some maybe not insoluble protein aggregated were formed actually. So when at a high as a high PEF strength, the protein will be aggregation again occurred among the proteins, and will be see, and then from this results we can know that for the PEF for the soybean protein, maybe just the medium field maybe and maybe have some improvement, but high field strength maybe have a bad effect to the protein, maybe cause the more aggregation. And from the aggregation force analyzed, maybe the densified the bonds, maybe the mean force in protein aggregations. And we can see that in the medium field strength can increase the SPI surface charge This is a very interesting results. And after used some maybe spectrometers analyzed the structure of the secondary surgery structures, we can see that PEP treatment has more pronounced effects on SPI surface structures. But for the secondary structures, maybe need a very higher field strength can change. And here I do a and very interesting. I think it's very interesting results. We can see that after withdrawing the PEF, the protein structures were particularly revisable, actually revisable, because we can determine that the fluorescence intensities. We found that after the treatment of PEF, the fluorescence will be generated decreased. And this generated decrease means that the structures maybe have go back to their original states, but not just I think maybe not complete to the original state. And maybe the structures maybe have some folded, but not have some folded, but not same as the original state. And we also use the Monica Dynasty's technologies used to simulate the kinetic change of 7S and 11S after the application of PEF stress. We can see that from the MSD and MSF, when the PEF stress very high field, the change is very, I think is a very big change, yeah, actually. And we found that we found that when with the increase of the PEF strength, showing a loss and disordered state, and the decrease of subunit pile structures unfolding was positively correlated with the PEF strength. And this is one mechanism about the PEF polarization. And another mechanism I focus on maybe from the free radicals oxidation. And I have detected the combine and the sulfur hydrogen groups in SPI. And we can see with the increase of the PEF strength, this content have significantly increased. And I think maybe it's because of the PEF induced electrochemical reactions that generated IOS or the radicals to attract the protein to make the protein and maybe change in the groups. And actually, maybe a highest and this generation of iOS maybe at a very high electric fields can get, and this will promote the conversation of free sulfur hydrous groups to disulfide the bonds. Actually, sulfur bonds may be formed the protein aggregation. Maybe from the aggregation, maybe the oxidant of protein accelerated the formation of protein aggregation. And this is a modification mechanism, as we can say. So I will not talk too much about these pictures, and it just means that at a medium field strength, they maybe have and maybe the aggregation behaviors will maybe not maybe decreased, and by a very high price, a higher field strength, maybe the aggregations will be enhanced. So this is the conclusions. The aggregation behaviors of SPI is sensitive to the electric field strength of PEF, and 10 and in the medium PEF intensities, maybe is attenuated the aggregation between SPI molecules, but where higher PEF intensity or strength maybe induce SPI intermolecular reaggregation. And ionization and radical oxidants by PEF promote the formation of the that in the protein aggregation may be needed to formed. About this one, that's disulfidated bonds between the protein maybe formed the aggregation between the proteins, and SPI structure change is response to PEF were reversible because after we withdraw the PEF, the structure will come back to their original structure. So this paper I have been published in for the hydrocolloid and to be the first authors. And we can see from the first paragraph I introduced, maybe we can say that the problem is the effects at a low field strength maybe have low modification effects. Just use the medium field strength and can reduce the SPI aggregation, but it's very limited effect. As we can see that the solubility is increased from 17 to 17.7. It's not very significant. But for high field strength, maybe promote the internal molecules aggregations of SPI. So we can see that for single PEF technology, maybe have a limit modification. So I want to share a new methods. Just use a PEF and pH shifting. pH shifting, what does it mean? It means that the protein forcibly will use the alkali treatment, maybe at a pH at maybe change the pH to 11 and then in this pH we use the PEF to treatment. After the PEF treatment, we retracement of the pH to 7. Because in food industries, we all know that the environment often in the natural pH maybe at 7. And after this treatment, I want to say the solubilities maybe can be further increased, or the aggregation may be changed, or maybe have and then maybe this method maybe have maybe is better than the single PEF technology. And here I need to say that that for the chemical regions, and maybe have some safeties for problems to the foods. But now here I change the pH in the 11, and many research change the pH in 11 and back to 7. The result is not good. But for me, I use the PEF to combination. The result is good. So if they want to get a very good results, they maybe change the pH to 12 or more, or 13 or 14. Well, maybe need more agent to the solutions, and maybe cause the safeties of the food. And I have detected the food safety is okay. It's okay. Just use this PEF and the pH shifting is okay. So just use the PEF and the pH shifting. Maybe have 3 methods. First, maybe have 3 different combinations. Firstly, we can just use a PEF followed by pH shifting. And secondly, we firstly use the pH shifting and followed by PEF. And the third is about the PEF combinated with pH shiftings. And so after the experiment, I found that the combination of the PEF combinated with pH shiftings have a very good results, and the solubilities will increase from 17% to 19%. And another results is also show a good chance to show that the aggregation of the protein will be destroyed, and the protein will have a very good solubility and functional properties. And based on these methods, I also do some research focus on some biological compounds to driving, efficient loading of lutein. Lutein is a very good thing, a good engine for our eyes. Yeah, for our eyes. So, but the problem of lutein is that they are bad. They are difficult to they are difficult in the water, and the solubilities of lutein is very low. And another problem is that lutein have a very it's very easy to after the thermal degradations. So we used these methods about the PEF combined with pH shifting these methods to preparate it of our SPI nanoparticles by these methods. And these nanoparticles can loading of lutein, and found that the loading affected of lutein was increased from the 55% to 77%, and can delay the thorough degradation of lutein. And this paper have been published in food chemistry, and I'm the first author of this paper. And the same as this research, I also focus on use the milk protein, BSA, and to enhance the building affinity of curcumin. This is very I know that you know that is a modern in the food industry because this compounds, this bioactive compounds is a very good to our healthy. So the problem is the same as lutein because they are poor water solubilities and maybe not good in the photo stabilities and the thermal stabilities maybe not very good. So not very well. So I use the BSA and with PEF to treatment of the BSA and to enhance the building affinity with this with these compounds and have good results. And this paper, and this is a very maybe at a not allowed to publication, and maybe at 2022. And this paper has been published in the food chemistry, and I also is the first authors. And this paper gets the highest cited paper in ESI. And as well know that if you I have talk about that the chemical SPI maybe have a very bad solubilities in the food industries, because they after a very high temperatures to treat it, and there will be aggregation or denaturation. So I also use this method, just use the PEF and the pH shifting treatment. We found that after the treatment, we can see that you can see this one. This one is just not used any treatment of this proteins. We can see that it's very poor in the solubilities, and the functional properties is very very low. So after the treatment of PEF and pH shifting and the combination of PEF and pH shifting, we can find the results. We can see the results in here. We can see the results is very good, and the functional properties have a very greatly dramatically increased from the 26 to 17. But only use the PEF, only increased from the 26 to 76. This is not good in this result. So we need to combine with pH shifting. And another functional properties also have a very high increased. You can see that the emulsifying and the forming of this SPI have a very high increase. And this one we comparative analyze the solubilizations of this SPI with the previous modified method. We can find that the previous modification method, we can find that our methods, I think maybe the time may be short, 8 minutes, but just use the US or HPP, maybe 10 minutes or more long times. But for me, 8 minutes is okay. And the result shows that 17%, not the high solubility, but I think maybe is okay. And this is the result. We use this methods in the food industries. In this SPI, just use in the food industry. But next time I use the SPI, just use the frozen dyeing, because we want to learn the structure change. But for the high temperature SPI, maybe they have they have deleted, so they not from their original structures. So I think first time I need to learn the structure change. So I chose the power from the freezing. And this paper were published in 2023 in the for the hydrocolloid as the first author. And this paper also gets the highest cited papers in ESI. So finally, I will talk about the perspectives of our research. And firstly, I think PEF is a promising technology for modification of plant proteins. And PEF combined with pH shifting technology can solve the problems of limited effect of single PEF modification. And the mechanism, I think, maybe focus on the free reading course formation, also or another, maybe the polarization. And I think this I want to talk because I try to detect the free reading course, but maybe for me I not get the suitable methods to detect the free reading coins. So I didn't get the results to detect it or the free reading coin. But I have saw Michael and your groups have do some research in here, and this is your group published papers. Yeah, I have saw this is a very interesting research. And I also want to maybe we can have collaborations from here and maybe know more about the protein and the free radicals generated by the PEF and what they will attract of the protein. These things maybe I'm not green and not unclear. So I want to solve these problems. And finally, I will share our groups. Our groups, we in Foshan, and Professor Zheng is my teachers. President is my teachers, and our team members have also Professor Zheng is one professor and associate professor and lecturers and so on. So Professor Zheng taught us President have do the research for PEF for 13 years. He taught us. So he want to do more research, maybe for 13 more years for PEF, actually. So because I'm young, so I will try my best to get more mechanism about PEF in plant protein modification. My English is not very well. That's all. Thank you for your listening. I hope you can understand what I'm talking. But if you can understand, maybe you can say the PPT. Maybe it's okay. That's all. Thank you.
Thank you very much. I really enjoyed that. I was really closely following each of the slides. So yeah,
Yeah,
so it was the slides were very clear. So although there might be some words I did not get, I mean, thanks to your clear presentation visually,
yeah,
it
yeah,
was very, very nice and very interesting results as well. I love combination of the both experimental as well as molecular dynamic simulation. So that's very, very interesting.
yeah.
So I would have some questions, but let me see if there are any questions from the attendees. So if there are any questions from the attendees, please feel free to raise your hand. Meanwhile, I'll check my notes and what. Okay, so if I may have a question regarding the your proposed mechanism. It
Yeah,
was it was one of the slides where you're showing like if you can go, I'm not sure which slide was there. If you go, it was the PEF modification mechanism slide.
yeah.
If you can go back to that slide, please.
Okay, okay. You mean this one?
It's slide 21.
21. Yeah, this
Okay, now I
here.
see it. Yes, this one. Perfect.
Yeah. This
So
one.
yes,
Mm,
exactly this one.
yeah,
Very good, very good. So Um, this is very interesting. I'm always intrigued when there is some non-monotonous change of the effect. So, for example, you've seen increase of solubility and then it decreases again, right? If when you are
yeah,
increasing
yeah,
the
yeah,
field
yeah,
strength.
yeah, yeah,
So this is
yeah,
very interesting. So I'm wondering for these 10 kilovolts per centimeter, where you see 10, 15, where you see increased solubility.
yeah. 15.
You said it could be because of the more negative charges, which would make sense.
Yeah, yeah,
But let me ask. So if you check when you check the zeta potential at of the sample treated by 10 kilovolts, did you increase? Did you see also larger zeta potential? When you had a look on I think here of the
yeah.
yes,
I think
yeah,
the charge change
yeah. I see.
here
Okay,
very
okay.
interesting, but here maybe the results I'm not sure. I just use the this method, use zeta potential
Yeah, yeah.
analyzed.
Okay, I
We
see.
found
It does increase.
yeah,
Okay, so
yeah,
it
yeah,
corresponds to the hypothesis. Yeah, it makes sense.
yeah.
Okay, interesting, interesting.
Just this is result, but I think here have many interesting research you can do because
Hmm.
just for soybean protein isolate is a very big proteins actually, but they have many compounds 7S and 11S. Well, in the future research I will focus on 7S or 11S, just this protein focus on the compounds of this protein. Yeah, I think maybe have more interesting results.
Okay, okay. So here with the if you can stay maybe on the same slide because you are showing this result microstructure. Because if you go back to this slide, this result microstructure, there was the one which you showed with the zeta potential and the reversibility.
Michael
Uh,
structure.
it's yes, this one, 18. Perfect.
This one.
Yes, correct. So what's also interesting, and we saw it in one of our papers, is the reversibility. So here, what's the data which suggests there is a reversibility? This is this fluorescence intensity versus time, right?
Yeah, yeah,
Okay, okay. So it's very mild, right? Because there is some variation.
yeah, 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 yeah. I think so.
So it seems to be there is some. So it means you collect the sample in the outlet, and then you measure the fluorescence. This is the fluorescence from, um, this is the intrinsic fluorescence, right? From the tryptophans and tyrosines in the protein.
Yeah, I know you mean. You mean that the Florence this machines is linked with the PEF equipment. You mean this? And actually, and I do this research. I PEF equipment near the machines. Oh, yeah. And after treatment, and I will give that uh, um, detected other fluorescences. But sometimes I think maybe we need to the results. Maybe sometimes I think maybe the result I have to 3 times or 4 times, yeah, and then you know that is very like this one. So the results maybe, and actually I think we need to do more research focus on this. And actually I think sometimes maybe not a very actually, yeah, because you know that I know you means, but it's difficult to change these two machines to combine.
Yeah, I
That
understand.
yeah, yeah. I saw your papers. Yeah, you have combine of PEF and fluorescing.
It's yeah,
Yeah,
it's a
yeah, I saw your paper.
chemiluminescence.
Yeah, it's very interesting, I think. So I also want to do some research from here, from these things, how
Yeah,
to detect it, the reliable of the protein of another protein, maybe BSA or
yeah.
SPI. Yeah,
Hmm. Okay, okay. So it's just maybe just checking again. This is fluorescence uh of the intrinsic um aromatic amino acids, right? This is the fluorescence
yeah.
of the. So if do you remember what was the excitation wavelength here? Was it the 280 or for this? What was the excitation wavelength for the fluorescence? You have it somewhere there.
Which mm, oh, sorry.
It was the
Which
same slide we went. There was a fluorescence intensity. There was emission spectrum. I was wondering what was the excitation wavelength there.
you mean this?
Yes, correct, correct. So there is this fluorescence spectrum which you showed.
Okay, sorry. Can you see? Yeah, it's okay. Just use the molecular dynasty the technology of this protein. Yes, this result I think the way you know that or high PEF strength vary. Yeah, and I have do the experiment with I found every times I use a very high electric field, this is very scientific. Yeah.
Okay, okay. So I was asking about the excitation wavelength in this fluorescence assay. Like what was the if in fluorescence you measure the emission spectrum, right? This is what you show. Yes. And what was the excitation wavelength? Was it 280 or 200 whatever?
Yeah.
If you in fluorescence, and did you okay, it's maybe not important now because I okay. Anyway,
Yeah.
I apologize. I'll need to finish because we have to go for another meeting in 1 minute actually. So
Okay.
thank you very much, Wang. It was very nice talk. I will
Yeah.
follow
Thank
up with
you.
you also to your professor. Will be glad to continue the discussion and exchange the information of pulsed electric field effects on proteins. So with this, I
Okay.
thank you very much
Thank you. Thank you. Thank
and
you.
take care, and we'll keep in touch. Thank
Okay.
you.
Bye bye.
Bye
Thank
bye.
you. Thank you. Bye bye.